So I have the pleasure of introducing Dr. Hasindu Gamaarachchi. Dr. Gamaarachchi earned his PhD in computer science and engineering at the University of South New Wales in Sydney, Australia. He is currently a visiting scientist at the Garvan Institute of Medical Research and a lecturer at the University of New South Wales. He has led efforts to optimize real-time nanopore sequencing algorithms and, my humble opinion, is a thought leader in the movement to make nanopore analysis open source and open science. And it is a pleasure to hear from him today. So the stage is yours.
Yeah, so as you know, nanopore sequences are pretty cool things, especially that I can have one at home and show it to you like that. It's a small sequencer. But of course, there's the Promethean, which is a large sequencer. But whatever it is, let's say, can you see what I'm drawing? Yes, right?
Yes, we can.
Let us assume now this is the nanopore sequencer we have. Okay, the cover. And here we have something called the flow cell that we load our DNA or RNA sample to it. And when you zoom into this, it is made up of an array of tiny little pores called nanopores. And when we further zoom into one of these pores, what happens is the DNA strand somehow go through this pore, right? And when that happens, the ionic current variation is measured using a picoampere ranged ammeter. And what we get as the measurement is called the raw signal, something that looks like this. Okay. And of course, the computer wouldn't have an analog signal. Therefore, this measured signal is in an actual digital form as a bunch of numbers. So the raw signal in the digital form. So internally, there's something called analog to digital converter that takes the sensor measurement and convert into a bunch of integer values. So that's how it works. And like a nanopore sequencer, we connect it to the computer using USB. And these raw signals are output in a format called POD5 these days. So nanopore sequences are made by the company called Oxford Nanopore Technologies today. So they output these signal values into a file format called POD5. So before POD5, which came, I think, a couple of years ago, the format was called FAST5. So it was a format based on the hierarchical data format, which is a complex format. But due to various performance issues that we showed, it's now retired. It's no longer there. So POD5 is based on what we call the Apache Arrow file format, which is a column-based file format. It's also quite complicated if you go into it. So today, therefore, we are going to talk more about the format called slow5, which we developed a while ago. And one of the reasons which is applicable at the moment is it's a very simple file format. We try to keep it as simple as possible while keeping the performance and storage also minimum. So it's very similar to other formats like SAM and BAM in sequencing space, making it easy for us to understand what's going on. So slow5, and the binary version is called blow5. To convert between POD5 and slow5 formats, you can use the tool that we developed called BlueCram. Okay, so the name of the tool is BlueCram. And anyway, once you have this like signal data, the next step is to convert this signal into the ACGT bases that all of you are familiar with, the DNA bases, or if it is RNA, the relevant RNA bases. So that process is called, have you heard the process names? It's called base calling. It used to be done using a software called Guppy some time ago. It's no longer there. Now it is called Dorado. So Oxford Nanopore Technologies name their tools using various names of fishes. So Dorado is a huge fish. Have you seen a Dorado? I tried to see what it looks like a while ago when it came, and it's a very big fish like this. And the software also big like this and complex. Okay. So yeah. So today we will instead use slow5 Dorado, which is like basically a fork of Dorado that supports slow5 format. So once we base call, we have the reads in a format called FASTQ, which is a very simple text-based file format. And then we can do various other analyses. For example, we can align these reads using software like Minimap2, written by Henley, one of the eminent scientists in the fields of bioinformatics, who wrote like also the BWA software and the SAM-BAM format you all may know. And when you align, it go into the SAM format, which is again a text format, and its binary variant is BAM. We can use SAM tools to convert between SAM and BAM. And after that, you can do things like variant calling if you wish, or like various tools like CLEA and like BCF tools. There's a lot of them. I'm not going to go into details. When you have variant calling, you get a format called BCF. And nowadays, these nanopore base callers are also capable of providing us modification calls like methylation, CpG methylation. So if you want that information, it's not possible to store it in FASTQ. So Dorado outputs in like unaligned SAM band format. And once you have that, you can still align, and then after having the methylation tags in your SAM and BAM, you could use various other tools like ModKit to get it into a like a TSV or a bedmethyl like text format. So likewise, there are quite a few other like downstream analysis that you can do. But today we are mostly going to focus on the first like few steps here. Okay, so now let me go into Google Colab. Have you used it before? You have? Okay. Yeah, I hadn't used it before, so for this I yesterday went and learned how to use it, and it seems like it's pretty cool. Yeah. Okay, so now let me create a thing. Let's call it Nanopore base calling. Okay. And here, importantly, I will remember to go and change the run type to GPU. The reason is I am going to do some base calling, and Dorado base calling is not very fast with CPU. Probably you will have to wake up tomorrow to see the results. That's why we put the GPU. Okay. So let me first download an example slow5 file. Okay. I have the link here. So we put this thing to say that it's a bash command. Going to download the file. And then this is a blow5. One of the pod5 files converted to blow5 already. So the signal from the sequencer is here. And okay, it's downloaded. So the next step is I'm going to install slow5tools so that we can see what's in this file. Okay, so let's go into the repository, and here I'm going to copy the Linux binaries, much convenient than trying to compile. Okay, so I'm going to wget again. I'm going to extract this file. In bash, when you say and, it means the previous command runs and after that the other command runs. Okay. Okay. Already done. And now what we will do is, let's see whether we have it. I think it extracted. So it should be slow5tools. Maybe we'll see a ls and see whether it's there. Okay, it's here. Slow5tools, 1 point this. First, it's a good practice to see if the file properly downloaded or it's corrupted. So I'm going to run a quick check command. It will see if the file is good, and if not, it will just complain. So what was the name of the file? It was chr. Okay, this. Okay, no complaints, all good. And next, let's do a simple command like slow5tools. I'll just, okay, now it's auto-completing. Good. Tools, stats. So that command will basically print like what's the compression format, the file version, and like how many reads are there. Here it's a very small file, only 117 reads. And then let's go into the view command to see what's actually in the file. We'll copy this, view, and put the name of the file. But here I'm going to put a head command because otherwise it will print the whole signal. And I did that mistake before, and the Colab got stuck. Okay, so I'm just putting a head therefore. Okay, as you can see, it prints the header. Now let me quickly go and show you the file structure. Okay, this is a very quick summary in a bit better formatted way rather than you have on the text here. Okay. So here, like we have this like header starting from the hash, which are like metadata for the whole file. Number of read groups, it's usually one if the data is coming from a single experiment. But if you have multiple experiments combined, this can be different read groups. But for simplicity, just focus on like one read group files. And then you have these metadata starting from the @ symbol. These are the global metadata values that are common for the whole sequencing run. For example, the experiment starting time, the flow cell ID, run ID, things like that. Okay. And then we have the data types of the like upcoming fields and the name of the fields, the read ID, read group, and digitization, things like that. So the read ID is a unique ID, and then these things, digitization, offset, range, sampling rate, they are per read attributes that are very useful for us when converting the digital signal back to the picoampere range. And then you have this length of the row signal, which tells us how many sampling points are there in this read. In this case, like 123,456 like values. And then we have the row signal, which is the comma separated set of values, long string. Okay. And then there can be multiple read group ones. So in that case, like see, like every metadata has 3 entries, one for each read group. And the read group field here tells which group it belongs to. Like you can have more, like a better look later when you have time. But yeah, so this is how you convert the metadata into the, this is how you convert the raw signal into the actual picoampere range using various metadata fields available for each read. So that's a quick summary of the slow5 format. So that's what we have here. So now let me do a tail to show the last signal read. Okay, as you can see, this is the read ID and various metadata fields and the raw signal. I think I clicked too early, so it messed up the output a bit. So let me rerun. Maybe I should just take tail-1. Otherwise, it's too long and it's a bit unhappy, it seems. So that will print only the last one. Okay. So the read ID, various metadata fields, and when we have the signal and so on. Okay, so that's what the file looks like. And now I'm going to show you some other useful commands that you can run. One of them is there is this subcommand called skim. So here it will print all the read metadata without the raw signal. That way our collab doesn't get stuck, because if you print the signal, it's quite long and large amount of text, so it can make the page go unresponsive. So here, as you can see, it shows everything else. Now I'll demonstrate how to do a simple bash calculation using a text file like this. What we would do is we will find the minimum length and maximum length of the raw signals here. Out of the 117 reads, what's the minimum length and the maximum length? For that, I will use this tool called data mesh. You may have heard of it before. So it's not by default here, I believe. Oh, I always forget this thing because I'm used more to the actual bash. Okay, it's not there. So what I would do is I will install it using sudo apt install data mesh. Okay. Now let me this slow5tools and skim. Okay, chart this. I believe it was the 7th column. So I'm going to do data mesh. I'm going to give a dash capital H because we have the header and we should ask it to ignore the header. The first line here is a header. This one. Okay. And then I'm going to say min 7th column and max 7th column. Or maybe we can put the mean value as well. Why not? Okay, so it prints 3 values. So the minimum raw signal length is 3000 this much. And the maximum is quite big. It's like 1.5 million. The average is like 310,000. So likewise, you can do very simple bash operations on slow5 files. Yeah, I have like documented quite a few bash one-liners in the slow5tools documentation. So you can have a look if you are interested. And yeah, okay, so now it's telling us, okay, we should be using the GPU. That's what exactly we are going to do. We are going to download Dorado, slow5 Dorado, and do some base calling. Okay, so let me download slow5 Dorado. I'm going to copy this command just to wget, download it, and I'm going to extract the file once it's, yeah. This is a big fish, so it might take a bit of time, okay? So we'll go back to our whiteboard just to like see where we are. So we are now doing this step. We downloaded the slow5 file, which I had already converted, and we use slow5tools to do some operations on the file just to see what it is. And then now we are going to base call using slow5 Dorado, and we expect to get a FastQ file, okay? Now it's still downloading. So
Can you maybe provide a little bit of insight into the conversion between the picoamp signal and the signal that's reported and the reason for that?
Yeah. So let me again go into that equation. So these like sequences internally to the connected to the nanopore, they have something called the analog to digital converter. So what happens is when the DNA strand pass through the pore, the sensor measures a value. But the analog to digital converter converts into a digital value. If it was like 10 picoamperes, it will be mapped to a certain like a integer. And what we get is these integer values. But in order to convert it back, there is a set of values given by the analog to digital converter, like the range and the minimum value. We have to shift it here and there and do that linear operation to bring back to the current level. So the equation is pretty simple. It's like this. So we have the raw signal value. That means the value which was provided to us by the analog digital converter. And there's this parameter called offset. We have to add it. And after that, we multiply by the parameter called range and divide by the digitization. And then you have this signal in picoampere range. Then only we can apply various signal mapping operations like the pore model we have, it's in picoampere. So yeah, that's the reason. So we have to do this conversion for every single read value. Like here we have like a few. Okay, let's go here. Yeah, see, like it's a very long signal. So per every single value, we have to do this conversion. And then you have the current in picoampere. I'll show you what this current looks like in a while when we base call and then we will visualize it. Okay, it's still extracting because it's like a 2 GB package. It takes a bit of time to extract. Yeah. So any questions so far while it's happening? Any places that I should re-explain?
I guess why would there be, I guess, differences in either the offset or the digitalization or the range variables?
Okay, so if we look at this example, you'd see that most of them are like very similar. Like read group, the digitization is all going to be same for a given read data set. But if you have a different sequence, it's going to be different. This is a Promethean data set. That's why it is 2048. But if you get a MinION, it's, if I remember right, it's 8,000 something, 8,192. So this is like 2 to the power, I think 11, right? Yeah, 2 to the power 11. So that means the analog to digital converter used has a resolution of 11 bits. But if it is a MinION, it's actually 13 bits. So it's a bit more granular. That's why it's 8,192. The reason in a slow5 why we have this repeated rather than in a header data is for convenience, because the signal is only useful if you convert to picoampere. So rather than the user having to go and find it in the header, we have it as a field here. Also makes it easier if you combine two data sets, like a Promethean and a MinION data set, much simpler to find it here. That's why. And the range, sorry, the offset can be different based on the pore. That's why it's different in each case. Okay. And then like sampling rate and stuff, again, going to be the same for a given sequencing run. Yeah. So did I like answer the question? Like the answer is like some of the parameters are going to change based on pore to pore and also the calibration. That's why we have different values for some cases. I think, yeah, it's done. Okay, so now let's see slow5 Dorado. There's a bin directory, not bin, and inside it we have slow5 Dorado. So I'm going to download a model now. If I remember right, the sub command is download, but I don't remember the exact thing, so I'm just going to print help. Okay, it's list. That will give the list of all the models here, quite a few. So I'm going to use the super accuracy, the best accuracy, because we have a GPU today. This one, because this is a latest R10.4 data set I downloaded. So that's the right model. Okay, so what I will do is I'll download -- model and give the name of the model and run it. Start loading. Okay, and now let's base call our slow5 file. So for that, we have to use the base caller command. Again, I'm putting help because by default, it outputs in SAM format. So we want to get it convert like in the FASTQ format. So I don't exactly remember the thing. Okay, so it's emit FASTQ, okay? So what I would now do is base caller. I give the model directory and our slow5 file, okay, and emit FASTQ, and that's it. And let's call it reads.fast and run it. As you can see, it detected the GPU and doing some like parameter benchmarks. And once that is done, it will like finish the base calling. Okay, CUDA 0. Shouldn't take too much time. So once it is base called, we'll like see what's in it, the head and the tail, and we'll see how many reads have been base called. So previously we did a slow5tools stat, right? And we found out how many reads were there. Okay, 117 reads. So we expect the base called result to have that many reads. Any questions? Meanwhile, it's running.
Do you want to discuss maybe some of the metadata in the slow five that you showed without the raw signal, but that was on the right?
Oh, yeah, the optional ones, right? Yeah. So, like after these like 8 mandatory fields, there are these optional fields, or the auxiliary fields, we call them. And these things can change depending on the type of the data. Whenever ONT provides update to the Minino software, they sometimes write newer metadata fields and sometimes remove them, sometimes rename them. So any of those come into auxiliary fields. One of them is this thing called the channel number. I told that the nanopore sequencer is an array of these pores, right? So basically this channel number tells you which particular pore it is. Not exactly, but very close. It's too detailed to explain, so let's say that. And then this read number means from a given pore, which read number this is. Starting from I think 0, say first read, second read, third read, and so on. And the start time in terms of the number of samples, and then there's this thing called the start marks. So actually, what happened is inside the a nanopore, if you go into one of these, it is called a channel, okay? But inside the channel, there are typically 4 pores and a thing called a multiplex, like a switch. So at the beginning, when you sequence, have you used Minino software before to sequence anything?
The students have not. No.
Hmm?
The students have not yet.
Not yet, right? Are you planning to use it?
Yes, we are.
Okay, so when you are doing the sequencing, you will see something called mux scan. So what it does is change the switch to every pore inside a channel and find out which pore has the best quality. And then it selects that. Okay. And if this is the mux position 0, 1, 2, 3, let's say I'm just taking example. Okay. The exact values may vary. So if the best mux was set to 0, these reads will contain 0 for that read. But later, after few hours, Minino software again does a mux scan, because these pores eventually get damaged and they die. So during the mux scan, they figure that out, and if it is a dead pore, it switches to the next best pore. So let's say now it's 2. So from that point onwards, the reads from that channel will get that particular mux value. So in a MinION flow cell, if I remember right, I think there is 500, approximately 500 channels. And each channel has 4 pores, so 500 into 4, around 2000 pores overall. Okay.
Right. Yeah, I guess I have the question. At this step, do we filter out any reads? Because Theo just said some of the reads have bad end reason, signal negative, I remember.
Yeah, so that is those kind of unnecessary reads are automatically filtered by the base caller if they are not good.
Oh,
Yeah.
okay. Okay, thank you.
And if necessary, you can even give a threshold, like pass threshold to the base caller. Then if the quality value is less than a given threshold, it will remove all those reads. Yeah.
Okay.
Can you give us some insight into how the base caller calculates those quality thresholds? What it's comparing to?
Yeah, that is done using a neural network. So you may know, like, these base callers nowadays, like, use deep neural networks. The most recent one, like V5 models, they use a transformer model, a very, like, a cutting edge neural network model. So then they build, like, break the signal into chunks, like, let's say 1,000 like signal points in one chunk, and then, like, send it through the neural network. And you get the output from the neural network, which is basically a bunch of matrix values, and that can be, like, decoded into the bases and the probability scores of how much the neural network thought that thing is to be correct. That's what we get as the quality score. Okay. Right, I think this is done. Okay, finished, good. So now let us go and see the head of the file. Okay, as you can see, we have the read ID, some metadata, the actual read, the plus sign, and the quality scores. Now let's see how many reads we have here. Okay, WC-L reads.fastq. We expect it to be how many lines? You know, WC-L prints the number of lines in a file. So fastq has 4 lines per read. So that means 117 into 4, 400 something. Okay. As you can see, it has been base called successfully. Now, what we will do is, we will try to visualize one of the reads using the squigualiser software, okay? But before that, we have to like, connect the signal into the base calls. Do some alignment of the signal to the actual base calls. For that, we will use the software called f5c. Let me download it here. Binaries. Very similar, like, steps as before for downloading the binaries. Okay. Now, let's see. Yeah, there are 2 binaries here, one for the CPU, the one with the GPU support. Here we have GPU support today, so why not? We'll use the one with CUDA, NVIDIA GPU support. So here I'm going to use this subtool called resquiggle. I have to give this thing. Let me see. Okay. So I will give the blow5 file and the base called read file as the argument. So this command will align the signal into the actual base call reads, okay? And I want the output in the PAF format, one of the formats to provide the alignments. Minimap, for instance, can provide those two for base calls. But here we are going to get the alignment from read to read. Let's call it resquiggled.paf. Okay, so it's running, detected the GPU, good.
Could you describe the PAF format?
Yeah, so the PAF format is a simple alignment format, pairwise alignment format. That's what it is, like, stand for. That was introduced by Heng Li, the Minimap author, some time ago. Now, if we, like, let's take the head of the file. So first we have the query ID, which is the read ID here. Okay, and the length of the read, the base code read. And then from this place of the read to this coordinate, it mapped to the reference, which is mentioned here. In this case, it's read to read mapping, okay? Signal to read mapping. So that's why this and this are the same. And then plus and minus, whether it maps to the plus strand or the minus strand. And then various other parameters. And finally, when we run using the f5c resquiggle, it gives some additional auxiliary fields too. This SS tag gives the annotation between the signal samples and the bases so that it can be used to visualize. So now let's download squigualiser to visualize these reads, okay? So again, I'm going to download the binaries and extract it. Okay, so it's downloaded a bit faster. Okay, good. So now let me run squigualiser. Let me ls, I don't remember which directory it is. Okay, just squigualiser. And there should be a binary called squigualiser. Okay, so now I'm going to use this plot command here. Okay, -f, we provide the FASTQ file. And then exactly remember, so I kept it written somewhere. Okay, anyway, so let me run without it, okay? Then it will print what arguments we need. Okay. Copy that part. Plot and -f is the sequence file, which is reads.fastq. And then I think -s is the slow5 file. Yes. And then we have to give which read we want to visualize. Otherwise, it will go and make plots for every single read, which is going to be a long time. So I think, yes, -r. And let's randomly pick one of these reads, okay? Read IDs. And then finally, we have to give -a, the resquiggled PAF format that contains the alignment information. You provide the base called reads, you provide the lo-fi file for signal, and the annotation between the two. And finally, the output directory, let's call it plot 1. Okay, it did the plot. Now we go into the file browser and we can download the HTML file and view it. I think it takes a bit of time. Okay, here plot. Okay, so this is what the signal looks like. So, okay, these red color dots are the actual samples, signal values, which was a comma separated set of values in the lo-fi file. And in the base calls, these are the bases and this is the KM. And here we see how it is annotated. Colors mean different bases, okay? G is yellow and C is like blue and so on. As you can see, like when the base changes, the KEMA changes, the current wave level like changes. But like it's very noisy signal, like it's not very obvious to the eye, but the neural network, once trained, can figure it out. Yeah. And also you see like sometimes like the bases are like having more samples than the others. This is one of the biggest challenges in nanopore base calling, because when the nanopore, when the DNA strand pass through the nanopore, you know, like there's something called the motor protein that try to control the speed, but still it cannot keep the speed at a like a same level. It slightly, you know, move here and there. The speed, it's natural. So that's why like sometimes when it is slower, you have more samples. If it goes fast, you have only like few samples. So it's like, you know, like I think of an analogy like a, you know, a leash dog passing through a gate, okay? You have a leash like the motor protein to control its speed, but still, you know, like you can't 100% control the speed of the dog, right? It can just go fast a bit or slowly. So likewise, the DNA strand also can vary like its speed, and therefore we have this problem. Yeah. And I even saw in recent papers, sometimes they even go like back a bit sometimes. Yeah. Anyway. Yeah, so that's what a signal looks like. Any questions before we move on to the alignment part?
I guess, well, so I guess my understanding of the nanopore is limited to their animation. So I would thought that like A would have the same signal and T would have the same signal, but here it is like very different.
I mean, the thing is the nanopore is like not that, you know, like narrow enough to constrain one single base.
Okay.
So at a time, there is a set of bases inside, which we call the KM. In
Okay.
R10 pores, like it's like 9 bases that affect the instantaneous current. So it depends on the KM rather than the base, okay?
Okay, so if like would the same two gamers have the same signal? Oh, okay.
Approximately the same signal. Here you could see, right? This, like whatever this KM here,
Mm-hmm.
have like kind of similar values compared to the others, right? It's
Okay.
a higher level, but it's noisy because this is a natural measurement. There are a lot of sources of noise, so you don't get a perfectly, you know, nice signal. You get a noisy signal like this. Yeah, but if you look at this KEMA and a KEMA of the same like value somewhere else, they should be approximately the same. Okay.
Okay, thank you.
So now let's do some alignment, okay? So in our chart here, now we did the base calling and we did an extra thing. What did we do? We try to get another color pen, okay? So we even like try to align them together using f5c resquiggle. And then we use squigualiser to visualize. Now let us take the FASTQ and align using Minimap 2 to create a SAM file. So for that, let me first download Minimap2. Fortunately, again, the binaries are there, so we can just download them. And I need the reference, okay? Reference genome. So this small data set is from the chromosome 22 of the human genome. I'm not going to map to the whole human genome because indexing it takes a while. So I'm going to just download chromosome 22 only, which I uploaded. Okay. Didn't copy. Okay, this particular location. Okay, downloaded. So now let's run the mapping command. Minimap2. Here we give -a to say that we want in the SAM format rather than the PAF format. And then we give the profile, map ONT, the type of data, because Minimap2 also can map other types of data like PAG, Bio, and Illumina. So here map ONT. And I don't want secondary alignment, so I'm just going to say no, okay, to keep it simple. One read, I need the best position, not like the second best and third best. And now here I'm going to give the reference. And then I'm going to give the reads FASTQ file, and I'm going to output it as read.sam. It's an amazing piece of software, like runs in no time, done. Okay. Now let's head the SAM file. The SAM file, just like a blow5 file, have these header values that are global to the whole file. Like here, like the command we used and the length of the chromosome to 22, things like that. And then we have the actual records, the read ID and where it aligned to, the cigar string that tells like how it aligns to each reference base and so on. Now, usually we need to convert the SAM into the BAM format, the binary variant, before doing most of the other analysis. For that, we use SAM tools. So SAM tools, you can just apt install. Done, right? Yeah, it's done. So now what I do is I will SAM tools, sort, reads.sam, and I can say reads.bam. Should be very quick. Done. And now let me create an index as well. SAM tools index. That's necessary in some future analysis steps to query based on a region of the genome. Like if you want chromosome 22's, this particular region, it can get those queries only rather than printing the whole file. So as the last thing, I will also show how we can visualize these alignments against the reference genome using squigualiser. For that, there are a few steps. I'll run through them very quickly. First, we will run f5c eventalign now to connect the signal file to the reference and the FASTQ together. Okay. Previously, we only combined the base call reads to the signal. Now we are trying to combine the base call read, the signal, and the reference together using 5c. So for that, we first will do f5c index, slow5 and chr5, and I'll give the reads.fastq. Okay, so I think it was Yeah, I think it was another name, right? Yeah, Linux CUDA. Yeah, I'll copy it so that it's easy for me. Okay, so now let me quickly run the eventalign command, okay? f5c, eventalign. The slow file with this, then the read file is fastq, and the BAM file is reads.bam, and then I ask it to output in the SAM format itself with the alignment information. And the genome is HG. Let's move it here, and then let's call it eventalign.sam. Move it here, run it. Yeah, it will take a little while. There any questions while it is running? I think we are running out of time, so I'll quickly show the next steps if it completes. Otherwise, I'll send you guys the notebook with the rest of the steps.
Can you talk a little bit about the banded alignment and the scaling calibration steps?
Oh yeah, so in the banded alignment step, what we are doing is we take the fastq read and then apply this hash table called the PO model. But this schema, this is the expected current level. We have such a table provided by ONT. So we take that to generate the synthetic signal. And then we take the actual signal and then align it using a dynamic programming algorithm called adaptive banded algorithm. It's you can think of it as a advanced Smith-Waterman algorithm catered towards signal mapping. And then once that is done, it generates some calibration parameters and finally send that to a hidden Markov model to get the actual event alignment. Okay, I think we are getting very close. And once it is done, we need to SAM tool sort the event aligned SAM file. Okay. It's the same SAM file we generated from Minimap, but it also includes this auxiliary field that tells this is how the signal maps to the reference. Yeah, okay, that's done. And now let me run squigualiser. The command is called plot pileup. Okay. And we have to give the -f, the reference, and then the signal file, and then the eventalign BAM file, and a directory, let's call it pileup. And finally, we have to give a small region. So luckily, I put the region I used for this small blow 5 here. So I'm going to put it like this, okay? And this is too big, so I'm going to make it slightly smaller contained. Otherwise, it's going to be a huge HTML file. Okay, we run it. Okay, I think I missed something. Signal 2. Let me double check the command. Plot pileup is a region. Yeah, I think, okay, so here I think I accidentally put the wrong region. Okay. Okay, so now it's generating the plots and you go to pileup. Oh, right, yeah, I renamed the thing pileup. So this is the right one, yeah. Thanks for that. Okay, so that's the right one. That's a huge one. So, okay, so this is what it looks like. So how for a given region, like a number of reads are aligned. You see, like, I mean, they are quite similar, multiple reads. If you look at the pattern, right? Yeah, and here and there we see some deletions, insertions, and those kind of things. Like this is how it looks like on top of each other. Not perfect, but quite close enough. Yeah. And some regions are pretty like messed up, the alignment. Yeah, and it goes on like that. And sometimes like we even can see like methylation in the signal itself. Can't exactly find a good region for it. But sometimes you see cases where here you see two clusters. That's when the methylation happens. Here it could be, see, like some reads are slightly higher level than this. So it could be some of them are methylated. Yeah. Anyway, I think that's all I wanted to like show from like up to like SAM BAM, and then also some visualization with eventalign. So a very quick introduction to signal level based calling and the rest of the steps.